Hello, and welcome to the virtual Teen Science Cafe, hosted through the South Dakota Discovery Center and our teen leadership team. And tonight, I am joined by one of our leadership members, Ray Starr, and by Dr. Victor Huber from the University of South Dakota. And I am going to have Race introduce our guest, and then we will have him do a presentation for you on vaccines and how our body responds to viruses.
Hi, my name is Ray Starr, and our guest today is Dr. Victor Huber. He's a faculty member at the USD Medical, if I'm right.
Mm-hmm.
And he's going to tell us about antibodies, vaccines. And I'll pass it on.
And actually, before you get started, I just want to let those of you who are watching know that if you type in your questions to us and comments, we will be able to answer those questions during or right after the presentation and potentially during the presentation. Okay, and with that, we'll hand it over to Dr. Huber.
All right. Well, thank you very much, Bree and Race, for the invitation, and everyone else who's organized the Teen Science Cafe. I'd like to thank you for inviting me to present to you this evening. As Race mentioned, I do work at the University of South Dakota, and I study virology and immunology in the medical school.
Before you get started, can you remove the split screen that you had, because we're not seeing your slide?
Oh, there's a split screen.
Well, you remember you said you had it set up so you could see both. So your slides are not showing up 100%.
They're not showing. Okay, right now, maybe if I go like this and then go full screen. How about there?
Perfect.
Is that okay? Sorry about that. So I'll be talking tonight about pandemics and vaccines and using influenza virus and coronaviruses as some of our examples. So let's start off with influenza viruses. Influenzas circulate every year. They usually infect during the winter months, and the symptoms of influenza include fever, cough, sore throat, body aches, chills, runny or stuffy nose, headache, and fatigue. And I'm sure you know someone or yourself have been infected with influenza virus, and it's a pretty nasty and aggressive viral infection that again circulates and infects people on an annual basis. The virus has its genetic material. This is a picture of the virus showing its genetic material on the inside, and two major surface proteins, hemagglutinin and neuraminidase. These are the proteins from where you get the H1N1 naming of the influenza virus. And those are the main proteins on the surface, and they'll be important for our discussion in the coming slides. So viruses require a cell to make more virus, different than bacteria that can grow on the surface of things like your skin. A virus must actually get inside of a cell. And so this shows the virus binding to a cell, going inside that cell, releasing its genetic material, and then turning the cell into a virus production factory. So basically, a single cell can produce hundreds or thousands of influenza viruses that can then go out and infect other cells. Influenza viruses exist in nature. They exist not only in humans, they also exist in birds and in pigs and in many other animal species. What this shows here is the movement of influenza viruses from birds into pigs, from pigs back into birds, and then from pigs into humans and vice versa. As you can see on this diagram, there are a number of different influenza viruses that infect birds, and there are a much smaller number that infect pigs and humans that have that ability to cross over into those different species. On occasion, influenza viruses can move directly from birds into humans, and these are those avian influenza viruses that have this infrequent movement into humans, but can cause pretty devastating diseases when they do that. So I'll present a little bit about influenza viruses and some of the pandemics that have occurred in the past. We're currently in a coronavirus pandemic. This isn't our first pandemic. It is our first coronavirus pandemic, but we've had other pandemics, including a few in the last 100 years that were associated with influenza. The first influenza pandemic, not the first one, but the one that we talk about the most is the 1918 pandemic, and it caused about 40 million deaths worldwide. This pandemic occurred before we even knew that influenza was caused by a virus. In 1933, influenza A viruses were discovered. In 1940, we discovered a second type of influenza virus known as influenza B. And in 1945, 12 years after we first discovered influenza, we created vaccines against influenza. 1947 was the first year where the vaccine did not protect against influenza virus, and this was the first example that the virus could change and vaccines would not work every year. So we had to update the vaccine on an annual basis. And in 1952, we established a network that does surveillance that sort of monitors the influenza viruses every year to update those vaccines. Our next major change in influenza occurred in 1957, and it caused another pandemic. We had a third pandemic in 1968 with another major change in the influenza virus. And in 1977, there was what's known as a pseudo-pandemic. It wasn't a true pandemic, but it saw the emergence of another H1N1 virus. In 1997, we saw the first avian influenza, the bird flu, moving from birds into humans. And in 2009, we had our most recent influenza pandemic, again caused by an H1N1 virus. The 1918 pandemic, as I mentioned, is the one that people talk about the most, especially now when we're in the middle of this coronavirus pandemic. This virus caused about 25 million infections in the United States and about 500,000 deaths. In England and Wales, 200,000 people died, and worldwide, about 500 million people were sickened by the virus, and estimates are anywhere from 20 to 100 million people died. Often they say about 40 million, like I said showed on the previous slide. When you look at the impact of this pandemic on life expectancy, this chart shows the change in life expectancy over the 100 years from 1900 to 2000. You can see our life expectancy moved from about 47 years of age up to about 75 by the end of this century. And you can see this significant dip in about 10 years of average life expectancy that was associated directly with the 1918 pandemic. So now that we know a little bit about pandemics, how do we end them? One of the ways to end a pandemic and to sort of return to our normal life would be the development and advancement of vaccines. So what is a vaccine? If you look in a textbook, you'll see a definition, something like this, where it says deliberate induction of adaptive immunity to a pathogen by injecting a dead or attenuated form of a pathogen. A lot of big words that basically mean we're trying to trick the immune system into thinking it has seen the virus or bacteria before. What we're trying to get from a vaccination are these antibodies. Antibodies have the ability to clear the virus, eliminate the virus from the host, and then provide memory so that you don't get infected with that virus again. On first exposure to a virus, you have a period of time where your immune system has to develop those antibodies. This is known as the lag phase. It takes about 7 to 10 days before you start making those antibodies. Those antibodies peak about 2 to 3 weeks after that first exposure, and then eventually start to decline. The second time you get infected with the same pathogen, you have a much more rapid antibody response and a much higher level of antibody, and you eliminate the infection much more rapidly. What we're trying to do with a vaccine is to mimic this response so that when you become infected with the actual pathogen, you can have a much more rapid antibody response, and you won't even feel that you've actually been infected with the virus. These antibodies work. The way these work are shown in this figure. As I mentioned before, viruses bind to cells and get inside those cells, turn the cell into a virus production factory. And antibodies basically bind those viruses and prevent them from infecting those cells. So let's shift over a little bit now into coronaviruses and where we currently stand in our understanding of coronaviruses and hopefully eventually developing a vaccine. There are 7 types of coronaviruses that are known to infect humans. The first 4 are associated with the common cold, and the other 3 are associated with the SARS/MERS family of coronaviruses. SARS-CoV-1 was the virus that first emerged in 2003-2004. It caused about 8,000 cases and 774 deaths. MERS-CoV-2 first appeared in 2012. It has caused about 2,500 cases and about 858 deaths. This virus does not transmit well between humans. It's usually animal to human transmission, so it's not been as big of a concern from a sort of human health standpoint. SARS-CoV-2, which emerged in late 2019 and is associated with the COVID-19 pandemic, has caused over 3 million cases worldwide, more than 220,000 deaths, and currently a death rate that's estimated to be about 7%, which is much higher than influenza, seasonal influenza, and even the influenza pandemic of 1918. That's a very aggressive virus that we're seeing infecting people, and it's shown here in this figure. SARS-CoV-2, looking at the virology, it's very similar. Well, it's very different than influenza, but it has a similar sort of mechanism of binding to a host cell, infecting the cell, releasing its genetic material, and then creating more virus. There's a spike protein on SARS-CoV-2 that binds a receptor known as ACE2, and that sort of initiates the infection process. So looking at vaccine prospects, what we want to probably do is try to mimic what we've done with influenza, create these antibodies against the dominant surface protein of the coronavirus. The dominant protein is known as a spike protein. If we can create these antibodies, we can block that receptor binding, we can prevent internal fusion, and have functions similar to the anti-HA immunity that we have with influenza. Some potential problems we'll have to keep in mind are the potential mutations of the spike protein and the fact that the immunity will likely wane like it does with influenza. So where we currently stand, we need to understand a little more about the virus itself and understand the immune response against it, and work toward developing a safe and protective vaccine. We have to remember it took 12 years to develop an influenza vaccine. And we're trying to create a SARS-CoV-2 vaccine or COVID-19 vaccine in about 12 to 18 months. And we're going to have to see how rapid we can do that. That'd be the fastest we've ever made a vaccine from the discovery of a virus to having an actual vaccine. And what we need to learn and understand is how the virus and the host interact, and look for those virus mutations and some of that waning immunity to see if this is a virus we're going to have to vaccinate against every year, every few years, or if it'll be one that we can vaccinate against once and have lifelong immunity. And with that, I'll take any questions or discussion that you might have.
All right, well, thank you. I will go ahead and start off with a question while we're waiting to see if we get some questions in the chat. Why isn't the flu vaccine as effective as other types of vaccines?
So the influenza virus, as I mentioned, with that change from 1945 to 1947, the virus itself changes very rapidly, and that makes it difficult. You develop immunity against it either from natural exposure or from the vaccine, and then the virus changes and escapes that antibody that's present. And we still, even after sort of studying it for 80 years, 70, 80 years, we still don't fully understand how the virus changes in a way that allows us to get ahead of those changes. We're usually sort of behind that and always trying to play catch up to the virus changes.
Okay. We do have a question in the chat, and it's, will the vaccine be safe?
That's always a question with vaccines, and it's something that we pay very close attention to. Even in the 2009 pandemic, when they developed that new vaccine, they did a lot of, when you got your vaccine, you actually got a card that told you if you had any sort of adverse events to call a number, and they would sort of track that is something they'll be paying close attention to, especially since it's a virus that we've never vaccinated against before. And that's why, the 12 to 18 months is a pretty aggressive timeline to create a safe and effective vaccine. And hopefully we can hit that mark. But we're definitely going to have to keep an eye out for that safety.
Okay. And then, so I'm just going to look at some of the questions that we have on here that are kind of related to this conversation. So I guess this kind of relates in terms of like looking at the efficacy of the vaccine. You mentioned that, like not knowing if there'll be something where we get it once and it's good for 10 years or a lifetime. Along those lines, why have there been so many struggles with the antibody testing?
So as a new virus sort of emerges in people, we don't know a whole lot about the virus. We do know that the virus has been mutating. We don't know what the importance of those mutations are. And so similar to influenza, if you develop an antibody against the virus, but you develop it against a part that changes, by the time you get that antibody test out, if the virus itself has changed, then the antibody won't recognize that virus anymore. Some of the antibody tests have been against more conserved proteins like the nucleocapsid rather than the spike protein. And it's kind of one of those things where we've got a lot of people developing a lot of tests, and it's going to be a while before we understand how specific and sensitive those tests are and can find one that's a truly reliable test.
All right, thank you. We do have another question in the chat, and that question is, why don't we hear much about the flu pandemics in the 20th century?
That's a great question. 2009, we heard a lot about it. It's one of those things that when the pandemics are happening or when we have a pretty severe influenza season, you'll hear back about them a little bit. But the 1918 pandemic in particular was one that was, it was right after World War I, and there was a lot of it was kind of lost for a while in the historical record. People really didn't talk about it. And actually, just a few years back, there were some books written about the 1918 pandemic that revisited that. But I think people see it as influenza. We're used to influenza. We have these sort of pandemics every 30 or 40 years, and then we make a new vaccine and it kind of goes away, and we're kind of used to that. This coronavirus was something that was brand new, and I think it's been obviously a pretty hot topic and a significant pandemic experience.
Hey, thank you. So while we're still talking about antibodies, can you explain how the plasma therapy is working that they're trying with patients?
Yes. So plasma therapy is taking the antibodies from people who recover from the infection and giving them to people who are actually fighting the infection. They actually did this back in the 1918 pandemic and have done that for a long time. The transfer of antibodies into sick individuals to try and help them recover from the infection is a really old practice. But it's a really effective one too, especially when you don't have a vaccine, because you can't help the person make those antibodies on their own. So the best way you can do that is to give them antibodies from someone who has actually recovered from the infection.
All right, thank you. And then there's another question in the chat, and it's what are the percentages of COVID-19 tests that are false negative, or is that an issue?
That's a really good question. As far as testing goes, I think the tests have been pretty good as far as they do a lot. So with the test that they're using now, instead of an antibody where the virus could change its protein and be different than that antibody, they're doing genetic tests. So if you can detect the genetic material, then it's usually a pretty effective test. It can detect even low levels of the virus. You're probably sometimes even a little more likely to pick up an early positive rather than the false negatives in that test. They haven't worked out pretty well, especially now that they've gone through. Early on, there were some issues with the testing, but they've worked those out.
Okay. Let's see. One other question I have is, so kind of going back to talking about the influenza virus, and you mentioned in terms of like impact to people and death rates being lower than with the COVID-19. How long do scientists think that the influenza virus has been a human virus? Like, how long has it been with us?
Yeah, so I showed the timeline started with 1918. That's one of the most popular pandemics to talk about. But we know the virus existed long before that. I think even the 1200s. I think I've seen even records that 800. There's potential evidence of outbreaks of viral infections that they assume are influenza. Some of the best records have been followed through the 1700s, which is when they identified that influenza could cause a primary infection followed by a secondary bacterial infection that led to deaths, and that's been well documented. So it's been around for a long time, and there was an 1890 pandemic that preceded the 1918 pandemic that was also well recorded.
All right, and then so along those lines, is talking about like the 2009-2010 outbreak. What I think is really interesting, and I wonder if you could just kind of share why this happened. Usually when we talk about outbreaks like this one, or even with the flu season, people who are typically at risk are really, really young and the elderly. But in 2009-2010, it was actually kind of an interesting situation because most of the people who were getting more sick were people like teenagers and people in their 20s and 30s versus the older population. Why was that so different?
Yeah, that's another excellent question. We also saw that with the 1918 pandemic, where the people in the sort of prime of their lives, the 20s to 35, were especially susceptible to the severe illnesses and deaths. And there appears to be some, as we mentioned, the influenza changes its type with these different pandemics. There's some evidence that earlier exposure, so in the 2009, they think that some of the exposure to the earlier H1N1s from the 1950s and 70s might have provided some protection against the 2009 influenza. So those older people were at a bit of an advantage because they had antibodies against those older H1N1s. In the 1918 pandemic, that 1890 pandemic, people who had lived through that actually had antibodies that protected them against the 1918 pandemic a little bit. And then and those theories are seen where the influenza virus, as it recycles and changes, you get this extended immunity in these older individuals that actually protects them more than you would see in a normal influenza season, where the elderly and the young are most susceptible to the severe illnesses.
Okay. Race, do you have any questions?
No, most of my questions that I had were asked already.
Okay.
Or I could ask how, because I've heard or I saw on the news that it was kind of hard to get tests out for COVID-19. Do you know like why that could be?
Yeah, that's an excellent question. They're still sort of working on that. With the emergence of a new virus, having it come so rapidly, as I mentioned, they do these genetic tests where they look at the genetic material, and that's how they identify the viruses in the sample. Well, in order to do that, you need these reagents, and some of those reagents aren't always readily available, especially if we weren't fully prepared for this level of virus circulation and need for these tests. So it became a bit of a race to try and make these tests and acquire the reagents. And it's important, the reagents had to show that they would definitely work. And so they have to run the tests and prove that the reagent works. And then if they're going to use a different reagent, they have to prove that that other reagent also works. And so that was some of the difficulty we ran into in the beginning, is that they hadn't validated some of the other reagents. So when they tried to switch them, they couldn't use that because it hadn't been validated yet. So they had to go through and prove that, a positive is a positive and a negative is a negative, and with the different reagents. And that was some of the sort of roadblock we ran into, and they've kind of gotten through that now. And now it's trying to just get those tests out so other people can use them and get tested.
Okay.
But part of that too is because it's a new virus that we weren't prepared to deal with. So.
Okay. That's the only question I can think of right now.
Okay. Just a reminder to those of you who are watching, if you have a question, you can type it in the comments on YouTube or Facebook, and we will be able to answer those questions for you. When we do these face-to-face, for those of you who are joining us for the first time, the vision of Teen Science Cafes is that they are opportunities for youth to have conversations with scientists, and it's led by youth and designed by them. And so we have a leadership team, and we invite any middle school, high school age people in the state of South Dakota, if you want to participate, to just get in touch with us through our Facebook page and or through our website, and we will get you involved. When these happen face-to-face, typically there's the presentation by the guest, and then there's a hands-on activity and probably some snacks and discussion. And so this has been kind of an extra challenge to be able to do this remotely. So we will close tonight with an activity. I think probably the easiest part will be that I will demonstrate the activity, and people can try to do it at home. And we would love to see you post pictures of the thing that you make, just so we can see what people come up with. But before we do that, there is another question in the chat, and it is, why does it take so long for a virus to get cured?
So the question about cured, within an individual, you have to develop that immune response and eliminate the pathogen. So again, it takes a little while to get those antibodies built up. The immune system needs a little bit of an exposure to develop those antibodies, such that then the next time you're a little bit more protected. On a global basis for developing those vaccines, again, the reason it takes such a long time to develop a vaccine against a pathogen, not only from the sort of vaccine testing standpoint, but you also have to develop and learn about the host virus interaction and identify what are known as correlates of protection. So with influenza, we know that when you get infected, 14 days later, you have antibodies that are assigned with protection. So if we can create a vaccine that mimics that antibody response, then we can have a sort of positive test and say you are likely to be protected against this infection. With SARS-CoV-2, we don't know that yet. We don't know what the antibodies look like after you've been infected. We don't know if you can be protected against reinfection. It looks like you can, but until we understand that dynamic of the host pathogen interaction, we can't really develop have that vaccine be both effective and safe.
Thank you. All right, so I am going to demonstrate the activity while we're waiting to see if there's any last questions. So the activity for tonight is to build your own virus. Viruses come in all shapes and sizes. So for example, the Ebola virus kind of looks like a worm. And then some viruses, like the coronavirus, is a sphere with all these proteins around it that give it that look like a crown, the spikes on a crown, which is why it's called a coronavirus. There's a special type of virus that goes after bacteria called a bacteriophage, and it kind of looks like this weird alien with legs. So viruses come in all shapes and sizes. They interact with plants, with insects, with bacteria, with humans, other animals, and do a variety of different things. Some are more harmful than others. And so the idea is that for you to kind of invent your own virus. So this is my googly eye virus, and it's just some pipe cleaners and little pieces of paper glued to a googly eye. And to come up with a name for your virus, and then to think about what part of what system or what group of organisms would this virus interact with in terms of either being a vector or infecting, and then what would that infection look like? So for example, if it were like a virus that attacked plants, does it cause the plants to not be able to process chlorophyll? Does it interfere with xylem and phloem and the movement of nutrients in the plant? Or is it affecting horses or deer or honeybees. So you can kind of think about all the different parts of our planet and what your virus would do to that system. And then just some other ideas of how you could do this. So I have some other found supplies in my house. So I just grabbed a piece of wire and I cut up some yarn, and I have an Easter egg left over from Easter, and I have a bunch of duct tape queued on the edge of my table. And my plan is to just kind of tape the yarn around the egg to be kind of like those protein receptors. And I'm going to figure out a way to kind of coil the wire around it too to create a different virus. So you can just grab a random supplies in your house and see if you can kind of create a virus of your own. And then we would love for you to post a picture on our Facebook page, or you can post it in the comments on YouTube, so that we can learn more about your creativity and your ideas about viruses. The videos are available to watch after we end the broadcast and share. So we please encourage everybody to share the videos and to please go to our website and check out more information about the Teen Science Cafe. And I do see one more question in the comments, and it's, do vaccines protect totally or make symptoms less?
That's an excellent question as well. So it's a little bit of both. So depending on the virus that you target with the vaccine, vaccine like smallpox protected against the virus pretty well, almost completely. And in fact, we were able to eradicate that virus from the planet, meaning that we don't vaccinate against it anymore, and we don't see any natural infections with it. With influenza, since the virus does change, there'll be a little bit less coverage every year, but even a little bit of coverage can help you replace the antibodies that aren't quite present and build that immunity. So if you get vaccinated, you may still get infected, but that infection is always going to be much less than if you hadn't been vaccinated. And sometimes you don't see the signs of infection. Sometimes you get a pretty minor illness. You can still go to school. You can still go to work. You don't feel as sick as if you wouldn't have been vaccinated and you got a full virus infection.
All right. Well, thank you. And I guess we'll go ahead and prepare to sign off for the evening. Brace, I just want to check to see if you have any new questions before we say goodnight.
No, I don't think I do.
Okay. So Dr. Huber, thank you again for joining us.
And thank you for the invitation.
And I will, if we get any more questions kind of posted after the videos are just out there, we'll make sure we forward those to you so that you can respond.
That would be great. Thank you.
Okay. And thanks to everybody out there who has been watching tonight. Have a good evening.
Thanks again.